Okay, welcome to chapter 8. We're now moving away from cellular respiration and into a process that is in many ways the opposite of cellular respiration, the process of photosynthesis. And in order to talk about photosynthesis, we're going to have to do quite a few things here. So we've got a little bit of our understanding of basic thermodynamics and metabolism already from the previous two chapters, but photosynthesis is going to introduce a few new concepts that, are like while they're tangentially related to the stuff we've already talked about, we're going to need to establish an understanding of how the excitation of electrons through light energy or from light energy is going to take place, because ultimately that's what's happening here in the process of photosynthesis. Plants and the other organisms like algae and bacteria that are capable of photosynthesis are utilizing light energy in order to form chemical bonds. They're taking energy from the sun and utilizing it to store energy in chemical bonds or in the form of chemical energy. So we're first going to look at the overview of photosynthesis, which is just generally what the process of photosynthesis is like. The next bullet in our lecture is going to be a little bit about the discovery of photosynthetic processes, and admittedly I cut some of this from the lecture in the interest of time, and also just because it's not particularly important. It's a little bit of historical information that just weighs down the chapter, and it's already a little bit of a challenging chapter. So we won't spend too much time talking about the actual history and discovery of photosynthetic process, but we will kind of wave hand at it. Then we're going to introduce a familiar concept, something that we've talked a little bit about already in the course of the semester. We're going to introduce pigments, and pigments are special molecules that can absorb light from the visible spectrum, and as a result, they can engage with light in ways that other molecules wouldn't be able to. And what I mean by that is pigments are capable of taking light energy and using it to excite the electrons in their molecules. So we've talked a little bit about pigments already, whether it be chlorophyll or carotenoids, but there's also pigments that we've used in the lab at this point. So if you recall, when we did the cabbage pH indicator solutions, we made those indicators with the purple cabbage juice. The reason that those change color was because they have special pigments in them called anthocyanins, accessory pigments called anthocyanins, that when introduced to different pH ranges change different colors. And so you have some experience with pigments already. Then we'll get into the trickier stuff with photosynthesis, namely the organization of the photosystems. So in plant photosynthesis, there are going to be two different photosystems, and they're responsible for different parts of the photosynthetic process. Roughly speaking, we can divide photosynthesis into two major parts: the light-dependent reactions, which are our next bullet point, and these are the reactions that, not surprisingly, depend on the presence of light. Now, plants can carry out the second part of photosynthesis in the absence of light, which is called carbon fixation. This is where they're actually going to take the energy that's been harnessed during the light-dependent reactions and utilize it in the process of taking carbon dioxide and converting carbon dioxide and water into sugar products. And I mentioned earlier in the semester that it's G3P, which can eventually be rearranged into glucose and sucrose and cellulose and all of the different plant parts that we're familiar with. And then we'll spend a bit of time talking about photorespiration and some of the different techniques that plants have discovered or hit upon evolutionarily in order to handle the problem of water loss or the problem of gas acquisition. And we've talked a little bit about this when we introduced the stoma or stomata in lab, the little pores on the underside of the leaves that are surrounded by guard cells. But we're going to look at how different plants in different environments are going to address the problem of water loss or address the problem of carbon acquisition. So again, photosynthesis is largely the opposite of cellular respiration. Remember, cellular respiration is taking glucose and um, glucose and oxygen, not necessarily water, although it does use a little bit of water. It takes glucose and oxygen, and it's going to convert those into carbon dioxide and water vapor. It's also going to produce an energy source, cellular respiration is, called ATP, right? Light and ATP in this equation represent the same sort of thing. Now, photosynthesis is the exact opposite. Instead of taking glucose and oxygen and making carbon dioxide, water, and energy, we're going to take carbon dioxide, water, and energy and make glucose, a little bit of water, and oxygen. So oxygenic photosynthesis is what we're really going to be focusing on, but there are many different types of photosynthesis, and some of them require some of them produce oxygen and others don't. Some of them require water, others use something else. Some of them use carbon dioxide as their carbon source. Some of them use something else. But again, we're going to be focusing on the basic oxygenic photosynthesis. And this form of photosynthesis, probably the most evolutionarily significant because it introduced massive amounts of oxygen into Earth's atmosphere, which previously wouldn't have existed in a molecular form. Our atmosphere is somewhere around 23% oxygen. And before the development of oxygenic photosynthesis by the earliest photosynthetic microbes, you would have seen molecular oxygen being vanishingly small in the atmosphere, insignificant quantities of molecular oxygen, and it would be unstable. But thanks to oxygenic photosynthesis, we have oxygen, we have ozone. It really enabled terrestrial life, it enabled aerobic respiration, and all of these other things that we take for granted. So cyanobacteria, which are bacterial cells that are capable of carrying out photosynthesis, can do oxygenic photosynthesis. There are several groups of algae that are capable of oxygenic photosynthesis, including the chlorophytes, the charophytes, the rhodophytes, and some in the Stremanopoles, the diatoms. We'll talk about those in Bio 112. And then the land plants are capable of carrying out photosynthesis as well. And the land plants are kind of special, and the ones that we're going to focus on, because they carry out photosynthesis in specialized organelles called chloroplasts. And you may remember that we talked about chloroplasts in the context of endosymbiote theory, the idea that some of the organelles that are possessed by eukaryotic organisms were actually once free-living bacterial cells that were engulfed by a larger cell and became symbiotic. Chloroplasts represent one such example, and in some of the algae, they're actually secondary endosymbiotes, but that's again something that we'll talk about in Bio 112. So, we divide photosynthesis into these two major stages, the light-dependent reactions, which not surprisingly require light. And the main goal of the light-dependent reactions is to take energy from sunlight and convert it into a chemical form of energy. They use sunlight energy to make ATP and also to make NADPH. Now, if you recall from our extensive discussion of NAD plus and NADH in cellular respiration, a molecule that, like NAD plus, takes on a hydrogen or dehydrogenates a molecule and an electron is going to be a high energy molecule. It's now carrying electrons that are capable of being passed down a series of proteins to form a proton gradient like we saw in cellular respiration. But NADP is a little bit different from NADH. NADP plus, although it does the same thing in terms of function, it's still an electron carrier, it still dehydrogenates something, and it still takes an electron from something or reduces itself or oxidizes the other molecule. It is a little bit different in its structure. Instead of just being NAD plus, it also has a phosphorus group. But otherwise, it's the same thing. NADP plus becomes reduced and takes on a hydrogen and an electron and becomes NADPH. And then that electron that it's carrying, also the hydrogen that it's carrying, can be used later on in the process. And we'll talk about how exactly it's used when we get to that. Now, the second part of photosynthesis is what we call carbon fixation, and this actually does not require light. So a lot of plants will do their light-dependent reactions mostly during the day, and then their carbon fixation more often at night. And this requires the carbon dioxide. You don't actually need carbon dioxide in the light-dependent reactions because you're not using the carbon to build carbohydrates yet, but you do need it during carbon fixation. And so if you remember during lab when I showed you guys those pores on the underside of the leaves, I talked about how a lot of plants will leave those pores mostly closed throughout the day, and then they'll open them up at night and they'll take in more carbon dioxide. We'll talk some more about the respiration in plants toward the end of the chapter, but the general idea is that the dark reactions or the light independent reactions do not require light, and so a lot of plants will do this at nighttime when there is no light. Many plants are going to be doing it continuously, but they'll do more of it when the light is absent. And the general idea here is that we're going to take ATP that we formed in the light dependent reactions, as well as NADPH, which again is a high energy electron carrier that we formed in the light dependent reactions. And we're going to use the energy that these molecules are carrying in order to synthesize organic molecules. And those organic molecules eventually get reconfigured into glucose or cellulose or starch or fructose, but the molecule that they immediately form is called G3P, and we'll talk a little bit about G3P later on. So this process, both of these processes, are going to take place within specialized structures in plant cells called chloroplasts. And chloroplasts are going to consist of a few membranes. They have an outer membrane, but they also have smaller structures inside called thylakoids. And these thylakoids have their own membrane, so they have an internal membrane system. Within the chloroplasts, you're going to find, and within the thylakoids, you're going to find the specialized photosynthetic pigment, namely chlorophylls A and B, but there are also some carotenoids or accessory photosynthetic pigments. The pigments are located in specialized structures within the chloroplasts called photosystems, and we're going to take a close look at the photosystems when we start talking about the light-dependent reactions. Now, the thylakoid consists of, or I'm sorry, the thylakoids stack up inside of the chloroplasts in these stacks of discs. They kind of look like little stacks of pancakes, and we call these stacks grana. It's plural, singular would be granum. And these sacs are suspended in a cytoplasm-like fluid, although it's not a cytoplasm. It's similar to a cytoplasm. It's a semi-liquid solution that the thylakoids are suspended in, and that's going to be important for the process of light-dependent reactions. So the leaf looks a bit like this. You have the outer cuticle, which is the topmost and thinnest layer, and that outer cuticle is going to be responsible for preventing desiccation or water loss. There's also an epidermis here, which is going to protect the plant tissue from abrasion or intense heat or sunlight. And then you have in this structure here the mesophyll, meso meaning middle. This region is where most of photosynthesis is going to take place. This is where the plant cells that can carry out photosynthesis, that have the extensive amount of chloroplasts, are going to reside. And a lot of this region is inhabited also by vascular tissue because the plant cells that are carrying out photosynthesis need to be delivered a supply of water and a supply of mineral nutrients in order to function. They also need a place to dump all of the products of photosynthesis so that they can be delivered and distributed throughout the rest of the plant, and that's what this vascular tissue is going to be responsible for. You can also see here on the underside of the leaf one of those stoma surrounded by two guard cells that we talked about in lab, and you can see here a cross section of the stoma, which again is going to be the pore that's responsible for permitting gas exchange with the environment. Oxygen goes out, carbon dioxide comes in. Water vapor is also going to vacate through the stoma, and this is one of the reasons why plants will only open up those stomata when they're actively in need of gas exchange. So each one of these mesophilic cells, or cells from the mesophyll, are going to have chloroplasts, and chloroplasts again are going to be the site of photosynthesis. You can see here they have multiple membranes. They have an inner membrane, or I'm sorry, an outer membrane, an inner membrane, but they also have these membrane-bound structures inside called the thylakoids. And the thylakoids, as we mentioned, are going to be the site of photosynthetic processes. This is where the pigments are, this is where the photosystems are, and this is where the proteins that are involved in the transport chain that we're going to talk about are located. So, as a general overview of photosynthesis, this is a pretty simplified introduction to the process of photosynthesis, but we're going to break down each one of these stages into a lot more detail. So sunlight reaches what's called the photosystem. If you look here, this is a thylakoid. This entire inner membrane system here is a thylakoid, and this region out here and up here is going to be outside of the thylakoid in the stroma. Remember that fluid-filled cavity that sits inside of the chloroplast but outside of the thylakoid. So sunlight is going to make contact with a photosystem, and when sunlight makes contact with a photosystem, it excites the electrons, which is something we're going to talk about here in a moment, in the pigment molecules. Those electrons get passed from pigment molecule to pigment molecule to pigment molecule, and eventually end up at the center of the photosystem, or a region called the reaction complex. Now, this is going to be energy that can be used to produce ATP down here and NADPH. And those high energy electron carriers and energy currency like ATP are going to be used in the Calvin cycle to take carbon dioxide and build bigger molecules out of it. So again, we're going to talk about each of these stages in more detail, but this is a general idea of how it works. You get sunlight, you use it to build ATP and NADPH. You use the energy in these short-term, high-energy molecules to build more long-term energy storage in the form of carbohydrates. So, as I mentioned, pigments are molecules that are capable of absorbing light energy in the visible spectrum. Now, we have to remember that light itself is a form of energy. This is why when you stand in sunlight, it increases your temperature. It's relaying some energy to your body. And some molecules are better at absorbing light than others. So light travels in these little individual units or particles or packets called photons. And when a photon makes contact with a pigment, it can release energy. And it can do so in a couple of different ways. Sometimes that energy is released as heat. So when photosynthesis, or I'm sorry, when why can't I think of this? When photons make contact with your skin, one of the reasons it raises your temperature is because most of the energy in that photon can't actually be absorbed. But in some molecules, they are capable of absorbing that energy. And what happens when they do is that an electron gets boosted into a higher energy level. Remember earlier in the semester where we talked about how if an electron is further away from the nucleus of an atom, there's more energy contained in that electron. And if it's closer to the nucleus of that atom, it has less energy. The electrons in a molecule that has been excited by absorbing a photon are going to be pushed further away from the nucleus, and they're carrying some energy. So plants have a wide variety of pigments, but the ones that are involved most in photosynthesis have a specific absorption spectrum. And what we mean by this is light is broken down by wavelength. and really all radiation is broken down by wavelength. But visible light exists in this range here, somewhere between 300 and 800 nanometer wavelengths. Blue light tends to be lower wavelength, which means there are shorter troughs. Red light tends to be higher wavelengths, which means you have a longer distance between the peaks and the troughs of the waves. Plants are particularly good at absorbing light from the lower wavelengths in blue and the higher wavelengths in red, but they're not especially good at absorbing light in between. And this is one of the reasons why plants are green. Plants are not green because they absorb green light. In fact, they're green because they reflect green light. They're absorbing the blues and the reds from the absorption spectrum, a little bit of the oranges from the carotenoids as well. But they're not absorbing the greens here at about the 550 mark. Those portions of light, those wavelengths of light are going to get reflected off of the plant tissue and not absorbed, giving them a green appearance. Now you might think, well then why are leaves in the fall red or orange instead of blue or green? And this is largely because in the fall, you start to lose some of the pigments that are absorbing the red and the orange spectrum. And instead, you're left only with pigments that are absorbing the blue and green spectrum. There is a little bit of green absorption in the carotenoids. This causes spikes in the orange and reddish color range. Reflection of red and orange light ends up taking place because nothing in the leaf tissue is absorbing those spectra. So there are a bunch of different pigments, as I've been mentioning, but the major ones that we're going to focus on are the chlorophylls, specifically chlorophyll A and B, though there are other types of chlorophyll, and the carotenoids. Beta carotene is a good example of a carotenoid pigment. And the carotenoids are also used in photosynthesis, but they have like an orangish or reddish color to them. So the chlorophylls are divided again into the two major groups, but there are more. Chlorophyll A, which is the main pigment involved in photosynthesis for plants and cyanobacteria. This is actually the only pigment that is able to convert light energy to chemical energy. Chlorophyll B is still able to absorb visible wavelengths of light and excite electrons as a result, but it can't actually convert the light energy to chemical energy. It can only pass those excited electrons, or the excitation of those electrons, I should say, from one pigment molecule to another. And this is going to be important when we start to talk about the photosystems. You'll see that chlorophyll B is responsible for collecting energy, but not converting it to chemical energy. That's only going to be chlorophyll A. So in addition to chlorophyll A and B, there are also carotenoids, and these carotenoids are going to largely be responsible for absorption of different spectra than the chlorophyll A and B, but they also have an important role as acting as antioxidants. So free radicals are particles that are moving rapidly around within cells. They're often products of metabolism, and they will oxidize things. So you don't necessarily want your proteins and your DNA to get oxidized by something. So you need systems that are going to protect them from oxidation. And carotenoids are often antioxidants that are capable of doing so. So the photosystems are organized into two major parts. The antenna complex, which is a bunch of different accessory pigments, carotenoids and chlorophyll B mostly, and these are going to collect a lot of photons and energy from those photons and pass the excitation or the excited electron energy, from one pigment molecule to another, all the way to what's called the reaction center of the photosystem. And in the reaction center, you're going to find the chlorophyll A molecules. So not surprisingly, this is the location where energy from sunlight in these photons is going to be converted into chemical energy, because remember, chlorophyll A, unlike chlorophyll B, is capable of converting sunlight energy, or the energy of excited electrons, into chemical energy. And we're going to see how when we start to explore the light-dependent reactions. So again, the antenna complex is going to be responsible for gathering all of the energy from the photons that are making contact with pigments and directing it to the reaction center. And that reaction center is going to contain chlorophyll A, not just chlorophyll B or other accessory pigments. So this diagram shows us this entire structure here, the blue structure, is going to represent the photosystem. Light enters the photosystem and makes contact with an accessory pigment. We're going to say this is chlorophyll B. Chlorophyll B passes that energy on to another chlorophyll B, onto another chlorophyll B, onto another chlorophyll B, and then another chlorophyll B. But eventually it's going to make its way to that reaction center, where chlorophyll A is going to be capable of passing that electron off, or I'm sorry, passing that excitation off onto an electron acceptor. Now, there's also an electron donor here that's going to provide a new electron to chlorophyll A every time it passes that electron on. And as you'll see in a few moments, in oxygenic photosynthesis, that electron donor is going to be a water molecule. And this is one of the reasons why oxygen is produced, because we're breaking apart a water molecule and dehydrogenating the oxygen in that water molecule. So the reaction center, again, is going to be largely chlorophyll A, and it's going to be capable of oxidizing the chlorophyll and then replacing its electron with another donor molecule's electron. So we have light. This is our photosystem reaction center complex. We have light that enters the system and it excites electrons. Eventually, that excitation is going to make it to chlorophyll A. The chlorophyll A's electrons become excited, they become higher energy, and they can be passed off to an electron acceptor. In this case, that electron acceptor is going to be NADPH, but once this electron acceptor takes on that electron, it is now reduced and the chlorophyll is oxidized. So temporarily, the chlorophyll is going to have a slightly positive charge, and this acceptor is going to have a more negative charge than it did before, although it will tend to be not charged anymore if it's NADP taking on an electron. Then the donor, the electron donor, which in the case of oxygenic photosynthesis is going to be a water molecule, becomes oxidized, and the chlorophyll then gets reduced, bringing the chlorophyll back to its original state, where it possesses an unexcited electron and it has a neutral state. It's not oxidized, it's not reduced, and it can repeat the cycle. Once again, sunlight enters the system and excites the electron. The electron in its excited state can be passed off to an electron acceptor. That electron acceptor takes off, leaves, and another electron acceptor joins up. The electron donor is going to dump an electron off onto the chlorophyll, and the chlorophyll is back to its original state. So that leads us to the light-dependent reactions, and this is going to involve the capture of light energy in 4 different stages. The first of which is the primary photo event, which is where the photon makes contact with the pigment molecule, and the excitation of electrons in that pigment molecule takes place. Then you're going to have charge separation, which is where the energy is passed along to the reaction center, and as we just saw on the previous slide, that excited electron gets transferred off to an electron acceptor. The electrons that get passed to an electron acceptor are going to go into an electron transport chain. And if you did your homework during the cellular respiration chapter, you're already familiar with what an electron transport chain is. Remember, it's a series of proteins that are embedded in a membrane that pass electrons from one protein to another, and they're going to use the passing of those electrons to release and capture small amounts of energy that can be used in different processes. In this case, the process is going to be chemiosmosis, which is also something that we talked about extensively in the last chapter, the production of ATP using ATP synthase and a proton gradient that's formed from the electron transport chain. So in many ways, this process is kind of similar to cellular respiration, but importantly, we're not breaking down glucose in order to extract energy. Instead, we're extracting energy from sunlight. We're using electron transport to form a proton gradient. We're using that proton gradient to produce ATP, and that ATP is going to be used not for cellular work, well, sort of for cellular work, but a specific kind of cellular work. Cellular work that involves producing carbohydrates from carbon dioxide and a few other precursors. So, oxygenic photosynthesis is going to involve two different photosystems, photosystem 1 and photosystem 2. One of those photosystems is going to be involved in producing NADPH. The other is going to be involved in producing ATP, and those are photosystems 1 and 2 respectively. Photosystem 1 is going to, again, take the electrons that are coming from the excitation of pigment molecules and pass them eventually to an NADP+, forming NADPH. This is going to be used to, later on, pass energy through the Calvin cycle and the dark reactions, or the light-independent reactions, to form G3P. Now, you might be wondering where we get new electrons for this photosystem after we use them. And the answer is we get electrons from the excitation of electrons in photosystem 2. It's a little bit counterintuitive because photosystem 1 actually happens after photosystem 2 does. The only reason we call them photosystem 1 and 2 is because photosystem 1 was the first one to be discovered. Photosystem 2 was discovered after that and therefore named photosystem 2, even though it precedes photosystem 1 in the metabolic process. So photosystem 2 is going to take a water molecule and oxidize it to replace the electrons that it's passing off to photosystem 1. And these two photosystems have a connection between them, which involve the cytochrome and B6 through F complex, which we're going to talk about in just a moment. So plants are going to use both of these photosystems, again, to produce ATP and NADPH for the Calvin cycle. And the electrons do not flow in a cyclical pattern through this system. The electrons come into the system and they leave the system. It's not like the citric acid cycle where something comes in and is reconfigured to produce the initial molecule again. It's introduction of electrons, utilization of electrons, the departure of electrons. So the entire photosystem, the entire light-dependent system looks a bit like this. Photons enter photosystem 2. The excitation of the electrons in the chlorophyll B molecules gets passed from molecule to a molecule until it reaches the reaction center. Here at the reaction center, a chlorophyll A has an electron get excited. Those electrons are passed to a series of proteins, plastoquinine and the B6 through F complex, which are going to create a proton gradient, much like the proton gradient that formed in the intermembrane space of the mitochondria during cellular respiration. Now, this reaction center, after it passes its electrons to the B6 through F complex, no longer has an electron, so it wouldn't be able to continue working. But what's replenishing those electrons is the oxidation of water. So we take a water molecule, we remove an electron from that water molecule, and we get rid of 2 hydrogen ions and 1 oxygen molecule. But it immediately forms a molecular oxygen in the form of O2, even though there's only 1 oxygen being lost from the water. So again, this electron is going to return to the photosystem and find its way to that reaction center, chlorophyll A, and then this process can be repeated. Okay, so the electrons that are excited get passed down this proton pump electron transport chain, and we form a proton gradient. The proton gradient is going to be formed on the inside, or I'm sorry, the elevated protein concentration is going to be formed on the inside of the thylakoid membrane, and ATP synthase is going to be embedded in the thylakoid membrane. As we've talked about extensively, ATP synthase is capable of synthesizing ATP by allowing hydrogen to flow down its concentration gradient. So the photosystem 2, again, is going to produce ATP before anything else happens in photosystem 1. Eventually, these electrons, which have lost a significant amount of their energy by being passed from proton to proton in the electron transport chain, are going to make their way to photosystem 2. The electron deposits into the chlorophyll A of photosystem 2, and photons are making contact with the chlorophyll B in photosystem 2. Those photons excite the electrons in photosystem 2, and they get passed to the reaction complex, chlorophyll A, which is going to excite those electrons and form an excited electron reaction center. The electrons from photosystem 1 get passed down through a series of proteins in order to reduce NADPH into, or I'm sorry, NADP plus into NADPH. So this is a really complicated process, but just speaking on it briefly again, photons enter photosystem 2, get passed, their energy gets passed from chlorophyll B to chlorophyll B to chlorophyll B. Eventually they make it to the reaction center where the energy is passed to chlorophyll A. The excited electrons in the reaction center get passed down a series of proteins. Those proteins pump hydrogen into the thylakoid. The thylakoid is then going to possess a proton gradient. That proton gradient is harnessed to synthesize ATP through the channel protein ATP synthase, and then the electrons from this system get passed to photosystem 1. The same thing is happening in photosystem 1 that happened in photosystem 2. Photons are exciting chlorophyll B. Chlorophyll B passes that energy from molecule to molecule till it reaches the reaction center and the chlorophyll A. The chlorophyll A electron gets excited, and that excited electron can be passed from protein to protein, and the energy from that process is used to reduce NADP plus to NADPH. This is complicated, I know, that's why I went through it twice. I would encourage you to watch this slide multiple times in order to make sure that you're familiar with this overall process. But generally speaking, photosystem 2 makes ATP by passing electrons down an electron transport chain, forming a proton gradient and permitting those protons to pass through ATP synthase. Photosystem 1 receives its electrons from photosystem 2, and that system is going to be responsible for passing electrons down a series of proteins that are involved in the reduction of NADP plus into NADPH. Okay, so as I just mentioned, photosystem 2 is going to involve the production of ATP through the excitation of electrons and the passing of those electrons down an electron transport chain, a series of proteins that pump protons into the thylakoid membrane. Photosystem 2, by contrast, is going to pass electrons in order to reduce NADP plus into NADPH. So here is that whole process on a more molecular level. You have the thylakoid membrane here and the stroma outside of the thylakoid membrane. Photons excite the antenna complex. They get passed to the reaction center. The electrons from the reaction center get passed to a series of proteins, plastoquinone being the first of them, and then some embedded proteins in the membrane. These embedded proteins in the membrane are going to use that energy to move hydrogen ions from the stroma into the thylakoid. So this is going to increase the hydrogen ion concentration inside the thylakoid and decrease the hydrogen ion concentration outside the thylakoid. So we get a gradient here. Now, as we know, nature hates gradients, so hydrogen wants to get back through to the stroma, and the only way it can is through ATP synthase. Using that hydrogen, like a hydroelectric dam spinning a turbine, we're going to produce ATP. But we're not finished yet, because that's just photosystem 2. Photosystem 1 then receives electrons from photosystem 2. Those electrons become excited and they can be passed along a series of proteins that reduce NADP plus to NADPH. Now we're walking away from photosynthesis, or from the light reactions, with NADPH and ATP, and you have a general idea of how exactly each of those is produced during this process. So the next portion of the photosynthetic process or photosynthetic pathways is what we call carbon fixation. And plants accomplish this in a number of different ways. Sometimes, as I've been referring to it so far, as I've been referring to it thus far, these are the dark reactions or the light independent reactions, where the energy that's been harvested from the light-dependent reactions, both in the form of ATP and through the reduction potential of NADPH. These molecules, if you recall, came from photosystem 1 and are then used to take carbon dioxide and build larger organic molecules that can be used for longer-term energy storage. The chiefly the production of G3P, which is a precursor to the carbohydrates that we know plants produce. So the most common way that plants are going to carry out carbon fixation is a process called the Calvin cycle. And this is named after the person who discovered it, Melvin Calvin. And we also refer to it as C3 photosynthesis because the first molecule that's produced as an intermediate in this cycle has 3 carbons. So C3 represents the presence of 3 carbons in that molecule. Now, what's ultimately happening here is CO2 is being taken from the atmosphere, and then that CO2 is attached to a 5-carbon sugar called ribulose 1,5-bisphosphate, and we'll just refer to this as RUBP going forward. This forms the molecule PGA, or 3-phosphoglycerate. But again, we'll talk about this as PGA. So again, we take carbon dioxide and a 5-carbon sugar called ribulose 1,5-bisphosphate, RUBP, and it forms 3-phosphoglycerate, or PGA. Now, there are a series of enzymes involved in these reactions, but much like in cellular respiration, we're not going to be belaboring the enzymes or the specific reactions that are taking place. We just want to have a general understanding of the process of the Calvin cycle, what the inputs and outputs are, and how they're interacting with one another. I do want you to be familiar with RUBP, PGA, or I'm sorry, CO2, where it comes from, atmospheric CO2, RUBP, which is joining up with CO2, and understand that the combination of those two molecules forms PGA, or phosphoglycerate. You can think of it kind of like how acetyl-CoA joining up with oxaloacetate forms citrate. It's a little bit like that, because now we're going to go into a cyclical process that's going to give us more RUBP, but it's also going to give us some of the byproducts of the Calvin cycle, namely G3P. So the phases of Calvin cycle are 3. There's carbon fixation, where RUBP attaches to carbon dioxide to form PGA. And again, fixation is taking atmospheric gases and putting them into organic molecules. So RUBP plus CO2 forms an organic molecule called PGA. We then have to go through a series of reductions in order to get PGA to form glyceraldehyde 3-phosphate, or G3P, and we'll talk about that in just a moment. And then we use G3P to regenerate RUBP. So RUBP can once again be used to join up with carbon dioxide and form PGA. Now, not all of the G3P we produce ends up being used to regenerate RUBP, and the extra G3P that we produce is used in the production of glucose or starch or cellulose or whatever it may be that we're producing. So this is a general idea, or I'm sorry, a general outline of the process. We have carbon dioxide joining up with RUBP. And again, notice the similarities to the citric acid cycle. We start with RUBP and something coming in from the outside. They join together to form phosphoglycerate, which is PGA. That PGA undergoes a series of reductions, and the process involves addition of phosphates from ATP that we produced during the light-dependent reactions. The phosphates can be then removed, and the reduction of this molecule will take place where we dump electrons and hydrogens from NADPH onto the 1,3-bisphosphoglycerate. And this eventually produces G3P, or glyceraldehyde 3-phosphate. Some of the G3P can then be sent off to produce glucose and other sugars, but most of the G3P is going to be sent back toward the regeneration phase, and we use a little bit of ATP to turn this G3P back into RUBP, which then can be used in the cycle anew. So again, RUBP joins with carbon dioxide, forms PGA. PGA gets rearranged and reduced in order to form G3P. Some G3P goes through the production of sugars. The rest of the G3P is going to be used to regenerate RUBP. Much like the citric acid cycle, in some ways it's the opposite though, because we were oxidizing citrate in order to get to the stage of oxaloacetate again and have the byproducts of the high energy electron carriers. Here we're reducing the molecule, and instead of producing high energy electron carriers, we're actually burning them up or utilizing them. So glucose, this is really surprising to a lot of people, because I think you probably learned in basic chemistry or in high school biology or whenever you last learned about photosynthesis, you probably learned that glucose is a byproduct of photosynthesis. But it's not a direct byproduct. In fact, no glucose is produced during the Calvin cycle at all. It's produced later on. We only get G3P, which is a 3-carbon sugar. This G3P is then used to make other types of sugars than glucose. We don't produce glucose directly. We're going to make disaccharides like sucrose or polysaccharides like starch. And each of these contains glucose. So sucrose is a disaccharide that has fructose and glucose in it. But we didn't make the glucose directly. We instead built a disaccharide that contains a subunit of glucose. Or in the case of starch, starch is a complex network and chain of glucose molecules, but we didn't make those glucose molecules individually. Instead, we produced a complex polysaccharide called starch. So photosynthesis is going to take the products from respiration and utilize them as their starting materials. So carbon dioxide, which is a byproduct of cellular respiration, is a reactant in photosynthesis. And cellular respiration is going to use byproducts of photosynthesis as its first reactants. So there's a really neat transactional process taking place here where the process of photosynthesis gives us what's necessary for carrying out cellular respiration, and cellular respiration provides what's necessary for photosynthesis. Now, the process of photosynthesis had to have preceded the process of aerobic cellular respiration because aerobic cellular respiration requires oxygen in order to take place. But there could have been other forms of respiration that were feeding into photosynthesis, just not oxygenic photosynthesis. So here's a general overview of the interaction between the mitochondrion and the chloroplast in a plant cell. You have the mitochondria carrying out cellular respiration, including glycolysis, pyruvate oxidation, and the electron transport chain. The final byproducts are carbon dioxide, ATP, and water vapor. Water and carbon dioxide from cellular respiration are used in photosynthesis in the light-dependent and light-independent reactions respectively. Because remember, plants don't use carbon dioxide in the light-independent state or the light-dependent stages. They use them in the light-independent stages. The byproduct of photosynthesis, which is oxygen and glucose, is going to be used to once again drive cellular respiration. So there's this really beautiful alignment of these two processes, and even though it only happens this way in plants, it still indirectly happens this way for animals because we produce a lot of the byproducts necessary for photosynthesis, and then our photosynthetic symbiotes or partners in life are going to produce a lot of the products that are necessary for cellular respiration. So it's handshaking, it's transactional. It's really a harmonious process. So photorespiration can be accomplished in a couple of different ways. And by this I mean the acquisition of carbon dioxide by a plant and the incorporation of that carbon dioxide into the organic molecules being produced by that plant. The first stage of photorespiration is carboxylation, which is adding a CO2. Remember, decarboxylation is removing a CO2 from something, so carboxylation is adding a CO2 something. Adding a CO2 to our UBP forms our PGA. And this is what normally is taking place within a cell. But the accumulation of oxygen can pose a problem for this process because CO2 and oxygen both want to bind to RUBP. When the photosynthetic process is rolling, you're producing oxygen, you're consuming carbon dioxide, and so sometimes the amount of oxygen is going to outnumber the amount of carbon dioxide. So plants have a couple of different answers to this problem. There are some plants that are just going to leave their stomata open all the time, and this permits carbon dioxide to flow in and oxygen to flow out. It also permits water vapor, however, to flow out, which is problematic for a lot of plants. So some plants can get away with this, but plants that live in arid conditions aren't going to be able to do this. They instead have to keep their stomata closed. Now, the problem here is if you keep your stomata closed, you're consuming your CO2, so your CO2 concentration inside of the plant tissue is going down, and your oxygen concentration is going up because you're producing oxygen. Now, this is going to interfere with carbon fixation because oxygen is going to more often then bind to RUBP, and then RUBP can't bind to carbon dioxide to form PGA, which is necessary for the rest of the Calvin cycle. So, C4 plants have developed a solution to this problem. C3 plants use Calvin cycle and C3 photosynthesis, but C4 and CAM plants are going to instead have an intermediate that's 4 carbon instead of 3 carbon. So instead of CO2 joining up with RUBP in C4 and CAM plants, CO2 is going to join up with phosphoenol pyruvate, which forms a 4 carbon molecule, not a 3 carbon molecule. Now, the reason this is great for C4 and CAM plants is because CO2 has a greater affinity, or I'm sorry, phosphoenol pyruvate has a greater affinity for CO2 than it does for oxygen. So it doesn't matter how much oxygen is present, as long as there's CO2 present, it's going to bind exclusively to the phosphoenol pyruvate, and oxygen won't be able to interfere. Now, this makes the process a little bit more expensive, but it also enables the plant to keep their stomata closed all the time or any time that they would lose too much water vapor as a result of having them open. Now, C4 and CAM plants both use phosphoenol pyruvate, but they do it in different ways. C4 plants have a spatial separation of these processes, and CAM plants have a temporal or time-gated separation of these processes, and we'll talk about each of those briefly here in just a sec. So, in C3 photosynthesis, in a mesophyll cell, you're going to get your carbon dioxide, join it up with RUBP, go through the Calvin cycle, and produce G3P. As a consequence, you have to have your stoma open a lot because you have to get rid of oxygen and you have to bring in fresh CO2. Now, by contrast, the C4 plants can hold their stoma open, but they compartmentalize these processes. There are mesophyll cells that are directly located next to the stoma, and these permit gas exchange, CO2 being acquired through the stomata, and oxygen being released through the stomata, but that CO2 is going to join PEP instead of RUBP. It's going to join phosphoenol pyruvate to form a 4 carbon molecule. That 4 carbon molecule gets shipped off to what we call bundle sheath cells, and these bundle sheath cells are not exposed to carbon dioxide or oxygen. Here, the process of photosynthesis takes place, or the Calvin cycle, I'm sorry, takes place, but instead of using that 3 carbon intermediate PGA, you're using the 4 carbon intermediate from the joining of phosphoenol pyruvate and carbon dioxide. The C4 pathway does solve the problem of photorespiration, but it costs more ATP to produce each glucose molecule. The cost is worth it though for these plants because they survive in hot or dry climates where they can't afford to leave their stomata open all the time. Now, another solution to this problem is what CAM plants do. In succulent plants, plants that store water like cactuses or the bromeliads like pineapples or just the generalized succulents, the ones that you could like purchase in Walmart and grow or fail to grow if you're myself or my wife. We kill all succulents. The stomata in these plants remain closed through the entire day, but they open at night. So during the day, the light independent reaction is taking place, and they're storing all the byproducts of the light or I'm sorry, I got that backwards. During the night, the light independent reaction is taking place, and they're storing the byproducts of the light independent reaction to be used during the day when the light dependent reactions are actually available. So what this looks like is CO2 is going to enter the plant tissue at night, and that CO2 is going to be stored in or fixated by PEP or phen oh my God, I'm sorry, I'm having trouble speaking. Phosphoenol pyruvate, stored by phosphoenol pyruvate in that 4-carbon molecule. Now you can't do much with this 4-carbon molecule during the night because you don't have the light dependent reactions producing your high energy electron carriers and your ATP. So instead you wait until day, and then at daytime you close off your stomata and you have all this C4 circulating, all of this 4-carbon molecule circulating. Then during the day, while your pores are closed, you convert the 4-carbon molecule to CO2 and run the carbon cycle like normal. So that's it for the photosynthesis chapter. This is a pretty complicated one, so I recommend that you watch especially the explanations of the light dependent and light independent reactions a couple of times. I'd be happy to answer any questions. If you have any, please feel free to reach out to me. And if you don't, I will see you later this week. Have a good week.